All right, I'm gonna go ahead and get started, largely because today I'm gonna talk about vaccines, and I could talk about this for like 4 days, and so I'm gonna try to keep myself sort of reined in, but I know I'm gonna need every last minute. I will also, just so you know, I have not yet posted the data from yesterday's lab. I have one other thing I want to try to do. I know I wasn't always explaining things super well because I was trying to think and explain at the same time. But basically, when you are doing cytometry, the assumption is that the whatever fluorescent thing you're using emits light at a very narrow band. It emits like just green and just at a very narrow amount of green. But in reality, sometimes it can be a little wider than that. And so the green thing could have like spilled over into the red channel. The dyes that we were using are both known as being pretty bad for spillover. And I thought the way I set it up was getting around that, but clearly it wasn't. I realized later yesterday that if I reanalyze the data on the software I used to use as a grad student, I might be able to clean it up. So I am in the process of getting a free trial of that software, which they're supposed to give me the key for later today. I'm just going to throw the data in there and see if I can get it any cleaner. If I can, then I'll give you the cleaner version. If not, then I'll give you the version you saw yesterday. So that's the story with that. And like I said, today I'm going to talk about vaccines. So we've already talked about secondary responses and memory when we talked about kinetics. And so you've already seen this slide where we can see how a secondary response is much more potent than a primary response in terms of speed of response after seeing antigen, in terms of magnitude of the response, all of those great things. And so as a result, generally the second time you are infected with some particular pathogen, you will not see any symptoms of illness because the immune response will be so fast and so rapid that it will get rid of the pathogen before you feel it. The idea with vaccines is largely the same. With a vaccine, we are attempting to give you some type of preparation, which is shown here, to induce a primary immune response so that if you ever are infected with the real pathogen, then you are seeing a secondary immune response instead of a primary response. And so you don't have much pathogen replication, you don't feel disease, all of that. And so basically what we're trying to do is figure out some way to make sure that you can get that primary response without having to get sick, without having to go through having the illness. If you remember the Biol paper where we talked about the dirty mice and their natural infections, realize that there is that downside of sometimes you die from natural infection. And so we want to give you a safe, sort of illness-free way to get that primary response so that with all subsequent infections, you're getting a nice secondary response. So these data are some data about the success of vaccines that are out there. So you've got a table as well as a graphic that give you information about number of cases pre-vaccine and post-vaccine per year and the % reduction. You can see that, like I said, in a graphic form. Here, the full syringe is the pre, the empty syringe is the post. And so you can look at this slide in detail. There is a slide that I'm going to show you later that has 3 links on it. I'm actually going to show you one of those links now because I feel like it fits in better with this slide. So if you really like this link that I show you, it's on the slide later with links on it. I love this little video. This video is awesome. In fact, the day I found it earlier this semester, every person who came to my office had to watch it. This one. All right, so what this is going to show us is it's going to show us data on vaccines over time. So So in the upper right-hand corner, we see the year. So you can see that this is going to start at 1912. And then you've got a list of different infectious diseases. And it's going to show you the number of cases of each of those infectious diseases in that year. If the bar is pink, that means that there's no vaccine available. When the bar becomes yellow, there's a vaccine available, though not many people get it. When the bar is green, most people are getting the vaccine. And so I'm going to let this play. You're going to see how many cases of these infectious diseases there are per year. And then once it gets up to, I think 2017 is where it ends, it will actually go back out and show you the max number in a year versus the current number because of vaccines. One thing I want you to notice is look at the x-axis and the number of million cases per year, because that is going to change a little bit throughout this graphic. That part is a pretty big part of this. So I love this graphic. So hopefully this showed you the success that vaccines have had in dramatically, and then other things happen, oops, in dramatically reducing numbers of infectious diseases. And so usually when I see data like that, my thought is go team immunology. And so if you think about the number of lives saved, the number of people who have been able to, go to work, the amount of economic productivity that's been saved because of people not being sick, all that stuff. This is pretty clearly the one of the largest, the most and largest public health intervention, most successful public health intervention that we've ever had. In fact, many of the big vaccines that we get were all actually first manufactured by the same guy, Maurice Hilleman, and he is generally cited as the person who has saved the most lives ever. There's documentaries about him and stuff. This is also some information about the financial implications of vaccine. So for every dollar that is spent on vaccines, we get $16 back, which makes it a much better sort of thing to spend money on than almost any of these other types of very useful things. And so if you look at that, again, vaccines are incredibly valuable. You can add to that things like agricultural vaccines and the amount of sort of productivity gained from agricultural vaccines, and realize just how useful all of vaccines are. Yep?
Agricultural vaccines as in vaccines for plants?
No, vaccines for livestock. So this is a list of a lot of vaccines that are available. Just because a vaccine is on this list in terms of being available or being a vaccine that exists does not mean that you necessarily get that vaccine routinely. So these are ones that are out there. However, the Centers for Disease Control has a specific list that it recommends that children get at particular times. And so this is the recommended immunization schedule for kids. here. So you can see that there are 3 doses of hepatitis B. You can see that the DTaP, or the diphtheria, tetanus, pertussis, is at a few different times there. You can see the polio vaccine. You can see the MMR. All of those things showing up nicely here. You will see, as I talk about some of the details about vaccines later on, some of the reasons why many of these have to be given more than once. And so one thing to note is that this is a subset of all of the vaccines that are available. Only the ones that are going to cause particular problems in children, and only those vaccines that are shown to be safe and effective are in fact recommended. Some of the others might be recommended if you are going to travel to certain places, or might be recommended by other countries. One thing, so I will also, because I'm me, talk a little bit about some arguments people make against vaccines as we are coming upon things here. So 2 of the different arguments, or one big argument against vaccines, comes up based on some of the data that we see here, which is people look at this and they say, no, giving my baby a whole bunch of shots at one time, that's too many antigens for them to be dealing with at once, or that's too many things for their immune system to deal with at once. So they're like, I'm not anti-vax, but I want to spread out my vaccines. And you will hear that a lot, that parents want to delay vaccines, but they still want to give them because they want to spread them out. So let's just think for a second, why are some of the vaccines scheduled at these times? What might be useful or important about these times that make this recommendation the way that it is? Yep, Mark.
So we learned that there's like parents and genes that get transferred to children. So probably especially the earlier vaccines are going to be given around the time where the parents' antibodies are starting to like degrade.
So generally those are moms because of the whole, placenta thing. So yeah, so we've got a whole bunch of antibodies from moms early on. And so if that vaccine was given too early, then mom's antibodies would destroy that vaccine before the baby's B cells were able to respond. And so generally we need to give these vaccines after mom's antibodies have degraded enough so that the baby's immune system will be the one that's able to really respond. Why else might we do this? There are 2 more things. Yeah, Rob? Yeah, babies are really susceptible to these particular diseases. These are, pertussis in particular is a good example, but all of these are really, really, really dangerous for babies. You kind of need to protect them ASAP. You don't want to like leave them around unprotected. You want to do something as early as you possibly can. There's one other thing. It's kind of tied to what Mark was saying. Yeah, Mark?
This is a long shot, but does it have to do with the fact that like when babies are younger, they make more gamma delta antibodies than alpha beta antibodies?
Well, so T cells, gamma delta and alpha beta are T cell types. So it's largely about kind of how able to respond or the types of responses. And so this is when we start to see sort of the right types of more adult-like responses. Mom's antibodies are gone, and we want to protect those babies ASAP. If you are delaying vaccines, you are kind of leaving your baby unprotected for a little while, which seems a little bit scary. How do you feel about the number of antigens that someone is being exposed to at any one of these vaccine delivery times? Yes?
Everyone's exposed to a whole bunch of antigens all at once. That's a dumb argument.
So when I asked that question, I went to stand under the air vent. I am likely being exposed to far more than that many antigens simply by the air that is currently blowing in onto me from this air vent. And so, if you are being exposed or challenged by 1 million antigens per day or 1 million microbes per day, adding 5 is not really a particularly significant change. If you hear those types of arguments, those are some of the things that we can think about with what we've learned about immunology to address them. So we can also talk about how vaccines are made. So successful vaccination depends on getting the appropriate immune response. As you'll see on the next couple of slides, just getting any old response is generally not enough. You have to have the right kind of response. So maybe if you make any old antibodies, it won't work, but if you make neutralizing antibodies, it will. Historically, this was done by trial and error. So Edward Jenner, when first making the smallpox vaccine, which is the first vaccine, did not know sophisticated molecular biology. He injected people and checked to see if they were protected. And in fact, he actually did a challenge and actually infected someone with smallpox to see if it worked. So it was totally trial and error. However, now we spend a lot of time thinking about the immunology, the virology, the molecular biology, all of that to try to make those vaccines safe and effective. In doing that, one of the first things we think about is something that is known as the correlates of immune protection. And so the correlates of immune protection can sort of be thought of in a simple way as basically what we do is we find some people who lived through the disease, or we find some people who didn't get very sick when they got the infection. And we do an experiment and we say, what kind of immune response did they make? And we say, okay, that must be the kind that makes you not get sick. Now let's figure out how to copy that in everyone else. And so we might say, oh, well, if we want to protect against chickenpox, we need some IgM, some IgG, and some CD8 cells. And if we want to protect against trypanosomiasis, then we need IgG. And so we might kind of study people who survived the infection, see what kind of immune responses they make, and then try to replicate them. And those immune responses that were made by the people who survived are known as the correlates of protection. So in some ways that makes sense and is pretty straightforward, but there are also some issues with that. I admit to being biased based on the paper that I'm showing you on the right-hand side. And the point of why I'm showing you this is that the reason why this paper was really interesting was that it was addressing a problem about correlates of immunity in HIV patients. We don't know of anybody who got infected and then didn't get AIDS. So we can't go ahead and look at them and see what kind of immune responses they made and copy them, because we don't know anybody who made those successful responses. And there are lots of different types of sort of more emerging infectious diseases where we have this problem, where we can't find the correlates in such an easy way. And so we end up doing things like using animal models and doing a lot of statistical analysis to try to identify those correlates of immune protection. Yep?
What about that person in the news who sometime, I think it was past year,
that would be known as the London patient, and we are going to talk about HIV the Monday after break, and I will go through exactly the therapy that that person received. One other thing that I will tell you about is that there are some cases where the vaccine-induced immune response and the correlates of protection don't add up perfectly for some vaccines we know about. So one vaccine that we know a fair amount about is the polio vaccine. There are actually 2 polio vaccines, and the one that I'm talking about right now is officially called IPV, or the Inactivated polio vaccine. But the point that I want to make you think about is that when you think about polio, what types of symptoms do you think of as being scary polio symptoms? Yeah. Paralysis. You think of it as sort of a neurodegenerative neurological disease. Yeah, Vanessa? Yeah, potentially leading to not being able to breathe because of paralysis. So then you might think about it for a second and be like, okay, so I mostly think about this as like a brain and nerve disease, but clearly that can't be the whole story, because I can't transfer a virus from my brain to your brain. I can't like think it at you. So there's got to be something else going on. And in fact, polio is in a group of viruses called enteroviruses. So you actually acquire polio by ingestion. It goes into your GI tract, into the mucosal surface. It eventually sneaks across the GI tract to a lymph node, replicates in the blood, and moves into the nervous system. It turns out that the IPV, the inactivated polio vaccine, which I will tell you, there are 3 different types of polio, type 1, type 2, and type 3. Within the past month or so, type 3 was declared eradicated. Type 2 had already been eradicated, so type 1 is the only one left, and this is largely because of this vaccine. This vaccine actually doesn't stop you from getting infected with polio. This vaccine induces antibodies that block the virus from being able to leave the GI tract. And so it just goes in and goes out and never is able to actually make its way into the nervous system in order to cause paralysis. And so what you can see is that sometimes the immune response that ends up working out for us in a vaccine setting might not be one that is the first one that you imagine. Sometimes, this vaccine is sort of one we did with trial and error, and it works. And we hopefully can eradicate polio because of it. Oh, look, there's an antibody blocking it. I didn't remember I had that slide. Another thing that is really important to think about when one is developing a vaccine is the route of immunization. And so we can think about giving vaccines either intramuscularly, so straight down into the muscle, subcutaneously, so under the skin, or intradermally into the skin. Different vaccines that you may have received may have gone intramuscular or subcutaneously. Patients often complain about vaccines and say, I don't like shots. I'm scared of needles. I don't want vaccines. Why can't you immunologists just figure out a way to make them all oral? So polio vaccine, for example, the other polio vaccine, OPV, is given on a sugar cube. You can also imagine ways that some vaccines could be given intranasally or things like that. So here's the dropper version. What kind of problems might you imagine with this? Yeah. Yes.
What's the difference between intradermal and
So subcutaneous is actually under the top layer of the skin. Intradermal is into that layer.
And then to answer the
Yeah.
question about problems, it's like not all of the antigen sensing is going to be from the GI tract. Like what if you need to have it go from the lymph to the blood?
So one question is about trafficking. Does it necessarily make sense to get your T cells primed so that they want to traffic back to the GI tract? Is that going to be the best way to, say, protect you against the flu? In that case, you want T cells going to the lungs. And so then maybe you think about ways to make an intranasal vaccine. Yeah, Paula.
About our stomach fluids, like the pH of it, like wouldn't it just destroy everything that's being delivered?
Yeah, so can we figure out a way to actually make it be stable in stomach pH is a problem? Yeah, Nick. Well, it's not sensitivity, but yeah, the other one. Non-sensitivity, tolerance. Yeah, so we could also induce oral tolerance. We know that oral tolerance is a big thing. And so, yeah, we could make it so that you are tolerant to measles now. That would be like even worse. You'd be not only not protected, but like extra allowing it. It would be bad. And so the route of immunization is actually a key thing that immunologists end up thinking about in terms of how to deliver these vaccines to deal with the trafficking issues and the oral tolerance issues, as well as sort of the anatomic issues like the stomach acid. And so this is a major thing people are thinking about. So there's actually another type of immunization that's really easy to do on animals. And so like we love to use it in the lab on animals, but the FDA would never approve it. And so we can't do that. there are lots of ideas about how doing intranasal could be really interesting and really cool, but I actually have done intranasals on mice and guinea pigs and rabbits, and in order to do it, you have to knock them out and then put the stuff up their nose, and there's a huge problem. The vaccines are almost never effective in animal models, and the reason is because they sneeze. And they just sneeze it out after you put it up their nose. And so you have to figure out a way to like inhibit guinea pig sneezes in order to develop some of these vaccines. Yeah.
What's the method that you use on animals that would never be able to?
IV or IP, both of them. Peritoneal or intravenous. So in order to do vaccine development and get a safe vaccine, you've got to think about safety. You want your vaccine not to cause disease and to have minimal side effects. You want to think about making sure that you have protective immunity in the population. So which antigen? Is that antigen going to work for every MHC type? Is that antigen going to work for all different people? Which type of immunity? Antibodies, T cells, whatever. Which route? You want that protection to be long-lasting. If you give someone a response and their immune response lasts for 2 weeks, and then they would have to come back to you 2 weeks later to get another vaccine, and they'd keep having to come back to you for every 2 weeks, that would not be particularly useful or a great idea. So for example, the hepatitis B series is 3 shots. I know some of you guys probably got it as kids, and so it wasn't as big a deal, but for the people who get it as adults, the percent of people who actually even go back for all 3 of them is pretty low. And I remember we were having a discussion in my PhD lab at one point. We were a vaccine development lab, and my boss asked us first to raise our hand if we got shot 1, and then shot 2, and then shot 3. And even among vaccine developers, like you could see a massive drop-off among getting all 3. So long-lasting protection is pretty key. You want that vaccine to be low cost. The World Health Organization really is interested in vaccines that cost less than $1 per dose. You want something that's going to be genetically stable. You don't want to be putting it in people and then having it mutate. You've got to think about the delivery issues, as I mentioned. And we also actually think a lot about storage considerations, and that is a huge deal in the vaccine world. So if you think about lab stuff, you can imagine, we always keep stuff on ice sometimes. Sometimes things need to be kept in freezers at -20. Other things need to be kept in freezers at -80, or even on liquid nitrogen, which is -236, I think. That might be the only way to keep your thing keep it stable, keep it from kind of expiring. And then imagine if you want to go around the world, can you keep a tube at -80 degrees Celsius as you move around the world? Are you going to have reliable electricity to keep your freezer on? Among other things. And so that's actually referred to in the field as maintaining the cold chain. And can you maintain a cold chain to actually store your vaccine correctly? So some types are actually way easier to store than others. And that might influence which ones people end up actually making. Yep, Paula.
Why might not a vaccine through like a skin patch work?
It potentially could. And so that is one thing that some people have tried to work on. The biggest issue is sort of how to get that skin patch so that it can actually deliver the vaccine where it needs to go without causing too many side effects. But that is absolutely something people are thinking about. Yep.
So what if someone doesn't respond to the type of antigen that's given if they don't have the right MHC to bind to the receptor?
It's a great question. I'm going to come to that in a couple slides. Mark, did you have a question?
No, I'll ask later.
Okay. So everything that we've talked about thus far is about what are known as is sort of this information about vaccines inducing immune memory against disease. And all of this is officially part of the individual effects of vaccine effects. And so if we have a person who's got their vaccine, they can be around some person who's transmitting, and they will not get the disease because they got a vaccine. Yay for them. And these individual effects are quite important. They allow us to mobilize the host immune system to prevent infection, and they're taking advantage of immune memory. However, vaccines have a second level of benefit. That second level of benefit is a population level effect. And so the population level effect also is going to actually break the transmission cycle of host-host spread. So it stops the infectious agent from spreading, stops the possibility of an epidemic. This phenomenon is known as herd immunity. And as you will see, the maintenance of a critical level of population immunity is really important. And so I've got a couple different ways to sort of show this to you. So normally, if we have a whole bunch of people who are healthy and say 2 people who are sick among that population, so these blue people are healthy but susceptible, and we have that infectious disease, we can see spread throughout the whole population. So basically, one sick person can spread to their friend, making their friend sick, and then we can spread to another person. And so even though person C might never meet person A, because of this chain of transmission, person C was getting infected because of person A. And so if person B had not been infectable, that would have actually protected person C as well. You can see this phenomenon here as well. So if we have an infected person, they may spread their infectious disease throughout the community. However, if a number of people in the community are immune, then we can see spread blocking. And some of these people, like person B, who never actually meets up with the infected person, will be protected at this point from spread. So here are the links. I'm only going to show you 2 of them because I already showed you one of them. This one is sort of a general thing that shows you herd immunity. And so what you can see in this is you're going to see red dots are people who are infected, green dots are people who are susceptible. You are going to sort of watch the infection move through the population. There are 6 columns. Those 6 columns vary based on what % of people got a vaccine. 0, 25, 50, 75, 90, or 95. So what you'll see as I push play is how this infection spreads in these populations. It'll make sense when it plays. And so you can see that infectious disease sort of sweeping the population pretty quickly with low percentages of people being vaccinated. But as we get to sort of a higher level of people getting vaccinated, then that will block spread. And this is important going back to Lexi's question. There are some people who get vaccinated and the vaccine doesn't actually work. And I'll mention that a little bit later. It might be because they have the wrong MHC type. It might be because that vaccine was designed to be given subcutaneously, or maybe it's designed to be given intramuscularly, and it was not delivered correctly. And so it actually went subcutaneous instead of intramuscular, and so it wasn't going to work. Maybe that person has some other immune deficiency. Maybe that person is on one of the many different types of immunosuppressive drugs that we've been talking about lately. Maybe that person has cancer. Maybe that person has an immunodeficiency. So maybe that person has some other infection that day that is sort of making their immune system somewhat compromised in making a response. There are lots of reasons why people might not have a good vaccine-associated response, and the way that those people are protected is based on everyone around them being vaccinated so that the infection cannot spread to them. And so herd immunity is the main way that we protect many people. There are people who are allergic to vaccine components. They are only protected due to others and herd immunity. Among many other things. In fact, with really small babies who are too small to be vaccinated, what you do is you vaccinate everyone who's going to come in contact with them. It's called cocooning. and make sure that they're not going to come in contact with people who are infected. And you can see that there is this pretty dramatic effect with herd immunity. My personal favorite view of this is here. This is a group from the University of Pittsburgh that has put together a simulator for measles. And it lets you actually look at whatever location you want. The closest one to here is Essex County, New Jersey, or Newark, New Jersey. And so I'm going to show you the data for Newark. So first, I'm going to show you the data on the left. And I'm actually going to sort of have it like this, even though it's a little small, because I think it'll make it a really, really helpful. On the left, when I hit play, you're going to see red dots appear, which are people who are infected with measles. And then as they recover, they'll become blue. You can see how many days have gone by at the top. And this picture at the left is going to show you what it looks like when 80% of people have a measles vaccine. Okay, so that is 238 days, so that's less than a year. And you can see how measles has swept through Essex County. And this is based on real population density data and all sorts of things like that. If you are not super up on your New Jersey geography, we are like here-ish. So like we're right outside this box. All right, so that's what happens with 80% coverage. Now I'm going to play what happens with 95% coverage. Because you were like, well, 80% is pretty good. But this is what we see with 95%. It's going to keep going out to day 238, but it's done now. So you can see the dramatic difference that 80 versus 95% has in terms of people having that measles vaccine. And so this is the power of what herd immunity can do. So the idea is that spread of the microbe is going to drop when the probability of infection is below a particular number. The exact number that you need to block spread of any particular pathogen really varies based on what microbe we're talking about. So there are lots of parameters that have to do with the microbe and how easily it's transmitted that vary here. So smallpox, 80 to 85% of people need to be vaccinated. Measles, it really needs to be about 95%. Unfortunately, no vaccine is 100% effective. And so even if 80% of people are immunized with measles, only 76% might actually be immune because of some of those questions about mystics or not having the right immune response, not having the right MHC type, things like that. And so you might be one of those 4%, but you're going to still be protected, and you'll never know if everyone around you gets vaccinated. If, however, everyone around you doesn't get vaccinated, you could get a nasty surprise. So I also want to talk about the types of vaccines and specifically how they work. So there are a bunch of different types of major vaccines that are out there. The first big type that exists is known as the live attenuated vaccine. So in a live attenuated vaccine, I am going to use words like live and dead today. And I know I'm talking about viruses, and I know we could have whole arguments about whether or not viruses are alive. We're just going with live and dead because it's easier than trying to talk around it. So yes, I know that I can sort of being a little fast and loose with some of the biology, but this is just easier. So we're using a live form of the microbe, but a form that has been weakened or attenuated in some way so that it is not able to cause disease. So it's going to cause either mild or completely inapparent disease. However, it will still replicate. All of the sort of parts of the pathogen's life cycle will happen. And so we'll get some type of nice immune response here. Some of the vaccines that you may have received that are live attenuated include the measles vaccine, the mumps vaccine, the rubella vaccine, and the chickenpox vaccine. So these are pretty great vaccines. In a lot of ways, these are sort of the gold standard. They induce super strong immunity. And that immunity usually lasts for a really long time. And it's sort of the most realistic form of the pathogen. All of the parts of the replication cycle are going to happen. You're going to get exactly the right cells being activated, exactly the right parts of the body. Like, you're going to get a pretty realistic infection here. But there are also some pretty big downsides, some pretty big cons to these types of vaccines. So what might you imagine would be big cons to getting a live attenuated vaccine? Yeah, Nick?
If it's live and able to replicate, you risk the possibility for genetic mutation.
Yeah, it could mutate and go from being weakened and attenuated and not serious to being a problem. There are some situations where you could imagine mutation back to virulence. And so that's a big issue. Yeah, Mark?
Can't that just be solved by mutating them?
Depends on what pathogen it is. Yeah, Vanessa?
I have just a question.
Yeah.
Like, how is it weakened and what essentially does weakened mean?
It's going to be the next slide. So in order to prevent this is one of the reasons why cold storage ends up being super important. So these are the ones where the cold chain is a really big deal. Because you're getting a natural infection, you probably are going to see some inflammatory responses. You are probably going to see a little bit of a side effect with this. It might be, a little rash, you might have a little fever, but this is the one where you're most likely to see those effects, which is good because it means your immune system is really working. But sort of these are the ones that are the most likely to have a bit of side effects, and they have the potential to mutate back to virulence. So Vanessa asked the question of how they are weakened. And there are two major answers to that question, which is sort of the old way and the new way. The old way is how a bunch of the vaccines that you have received were really weakened. The new way is how we weaken new ones that we are trying to develop. So the old way is you take the microbe, you put it in culture with some cells. In this case here, they're showing that you put it in monkey cells instead of human cells, so it's like a little bit off in the type of cell that it usually is infecting. And then you wait. And you wait for it to mutate. And from time to time, you take some out of your culture and you check and see if it still is causing disease. And eventually, you get some that has mutated and is not causing disease. One thing that you also might realize here is if, say, you were a person who was immunocompromised, one of these vaccines could still be pretty dangerous in you because it's replicating. And that's actually a big problem that we see with the live attenuateds, is that people who are immunocompromised are unable to get those vaccines. So like I said, you wait. It's kind of random mutations. We just sit there and hope that someday we're going to get a weakened version that doesn't make people sick. In the case of the tuberculosis vaccine, which I highly doubt I will have time to get to today, they waited for 23 years. So before they got a mutation. And so it was just sort of a random wait process. Now we actually do molecular biology and microbiology. We find the genes that are involved in virulence, and we either take them out or mutate them actively in order to design a weakened type of the pathogen. Yep, Lexi. Yes, exactly. So we are specifically choosing mutations or genes to delete. Yep.
So the second case, it would be easier to kind of take out multiple genes
But-
and kind of prevent.
Potentially. The thing is, you've got to get it so that it will replicate. But and there are some case studies I can tell you about where that seems like a great idea, and then disaster struck. The next sort of big type of vaccine, if you were to say go ask a layperson about vaccines, they might be able to tell you that a live attenuated is a thing, and they might be able to tell you the next one is a thing. They probably won't be able to tell you any of the other ones are. So with the other big type that people know about are the inactivated vaccines. And so here we've used a killed or dead form of the microbe. So now there's no replication present, but maybe some of the antigens are still present. Sometimes they're denatured, which might be bad for antibody responses, but T cells don't care. And so it might be okay in terms of antigen presentation on MHC, for example. You still might get mild or inapparent disease, but it's a lot less likely because you have no replication of the microbe. If you get a flu shot, so the shot version of the flu vaccine is an inactivated. If you get flu mist, the one that goes up your nose, that one's actually live attenuated. If you have gotten the polio vaccine that you probably got was also an inactivated. So if you think about this, what might be a good thing about doing your vaccine as an inactivated? Yeah?
There's no chance of the virus replicating and killing you, especially for immunocompromised people like me.
It's going to be way safer, especially in immunocompromised people. In general, it's way safer. You're not going to have to worry about mutation, so it's going to be pretty genetically stable. So all of those things seem pretty good. What kind of problems might you see? Yep. It's going to be way weaker. You probably aren't going to get all the steps of the replication cycle. You may not get all antigens produced. And in general, you're going to see a weaker response. And I'll say a little bit more about that in a couple of slides. Yeah, Vanessa?
How does it induce a mild or an inapparent disease instead?
Because it still might induce some inflammation.
Just that it finds it?
Yeah, I mean, at some point you're still stimulating PRRs, you're still making inflammatory cytokines. You can also imagine that, of course, this relies on you to be able to inactivate your microbe successfully. Also, there are some people who are against vaccines because of the inactivation methods. So for example, sometimes we inactivate using ultraviolet light. So we'll hit the microbe with UV light. It's not then UV for the rest of its life. Like if I go out in the sun, then I'm not going to give off UV to you. Similarly, if we are treating that with formaldehyde, you wash the formaldehyde off after you've killed things with it. So there are some people who are like, no, I don't want to use these because they have these harmful chemicals in them, but you do wash those off. But then one of the places where we've really started to design new types of vaccines are in types that are known as toxoid, subunit, and conjugate vaccines. These all have certain things in common. And in fact, you know enough immunology to understand both what they have in common and their differences. Yay, you. So in all three cases, a toxoid, a subunit, or a conjugate, we are immunizing not with the entire microbe. We are immunizing with a purified component of the microbe. So we might take the microbe, break it up in the lab, get out a whole bunch of our favorite protein and use that. We also could just make our favorite protein in E. coli and never even have the microbe in the lab and make boatloads of the protein that way. With a toxoid, we are using a toxin from the microbe. And so really what we're trying to get is antibodies that will neutralize the toxin. The toxin is a protein. So your diphtheria and your tetanus vaccines are both toxoids, where we're giving you a toxin and having you make antibodies that will neutralize. The conjugate is a carbohydrate from the microbe. And so many microbes have unique carbohydrates, unique polysaccharides on their cell surface. A conjugate, you might realize, cannot just be the polysaccharide. It has to be bound to some other protein as a carrier. It must be conjugated to a carrier, which is why it's called a conjugate vaccine. And so there are some cases where you take some kind of polysaccharide, you link it to a protein. Why they always want to link it to a toxoid in these slides makes me angry. But we're going to link our polysaccharide to some kind of protein to get internalization and to get that linked recognition B cell response. But again, we're just using a purified component of the microbe. Alternatively, we can use a subunit vaccine. Now, again, we are going to use a purified component, and this at this point is generally a protein. So we don't have to worry about, putting it with some other types of molecules. We could do it by breaking up the virus and just taking the protein. We could also make that protein through molecular biology techniques in the lab and never even have the pathogen in the first place. Just so you know, if you've received the hepatitis B or HPV vaccines, those are both subunit vaccines, where one protein from the outside of the virus is used to induce an antibody response. If you look at toxoids, subunits, conjugates, what can you think of as being pros of these vaccines? Yeah?
They're probably easier to make, right? Because, like you said, you can put it in
Yeah. So now that we're like good at this, they're not too hard to make. There's one huge pro of these. Yeah.
Everyone's responding to the same antigen, so it's easier to know the
Okay. So still, it's so big. There's one such big thing about these. That's a pro. No chance of infection. These are crazy safe. The safety profiles on these vaccines are amazing. Because you don't even have the microbe. In some cases, you didn't even have the microbe in the lab. There is no way you're getting infected. There is no way you're getting sick from these. And so safety-wise, these are amazing. I will tell you that when I first saw the data on the HPV vaccine, I was like, are you kidding? Because it was such beautiful data on how nice it was. And so these are all really, really, really safe. The problem is that you may or may not get good antigen presentation. And in fact, inactivateds and conjugates and subunits and toxoids all share one other big problem. None of them replicate. They might not stimulate PRRs. If you, for example, inject with a purified hepatitis B protein, there's nothing that's going to induce inflammatory cytokines and hit a PRR. You're going to have, turn on B cells and T cells, but you're not going to necessarily turn on any PRRs and get inflammatory cytokines, signal 1, signal 2. So you're in bad shape. Yeah.
Why not just do what you did with the problem with the last one?
We're getting there. There may be issues in how these guys access antigen presentation pathways, but the replication is a huge thing. If you use a live vaccine, you put in some dose, and it replicates and it amplifies, and that pushes up a nice high immune response. If you use one of the types that doesn't replicate, you put in the dose, and that's what you got. And then you might, in order to get that immune response up to a really high level, have to give multiple doses or multiple boosts. Most of those vaccines that, if you look back, that have multiple administrations, multiple boosts, are some type of inactivated or subunit vaccine that does not replicate and thus does not self-amplify when it is delivered to our patient. So what we can do in these situations is we can think about how can we add or give some type of PRR stimulation. And so very often we have to give an additional molecule, like I did in the problems, which is known as an adjuvant, in order to help out these vaccines. And so these vaccines also require adjuvants, whereas the live attenuateds may not require an adjuvant to be delivered with them. adjuvants are substances that stimulate early processes in immune recognition, especially PRR stimulation. And so the idea is they will actually help you get a better immune response with less antigen. There was this one vaccine we were working with when I was in grad school, and we were looking at sort of like how much immune response you got in a mouse and how much we wanted in a human, and we tried to calculate how much we would have to give a person to like get the immune response we wanted, and we calculated that if you gave the person the mass of an elephant, then it would be enough to get the immune response we wanted. I am not going to inject you with the mass of an elephant of anything. And so we were like, okay, clearly you need an adjuvant so that we can use less antigen to get a response. Adjuvants can work in a few different ways. So some of them are really sort of oil. It's more about them being oily and sort of acting as oils. And so sometimes what they do is they actually just help keep the antigen stable. They keep it from getting degraded so it sticks around for a longer time. Or they can actually directly be PRR stimuli. And most of the adjuvants that are under development are acting as PRR stimuli. And that's one of the reasons why PRR biology is very hot in the pharmaceutical industry. What you can also imagine is that adjuvants are actually really tricky in terms of study and with the FDA. So I'm going to go to this slide, and then I'm going to come back for a second. So this is sort of a list of adjuvants that are out there. Some of them you can see are oil and water emulsions, and they're really more about stabilizing the antigen. Whereas in other cases, we have things like some type of something that's going to actually stimulate a PRR, like nucleotides or things like that. But what you can imagine is that because these adjuvants are frequently stimulating PRRs and inflammatory cytokines, they stimulate inflammation. And so they make some of the vaccines a little side effecty. And so it's really hard to sort of get a vaccine approved with adjuvants and to say, no, those side effects are okay. And so there's sort of this big fight about what is and is not sort of appropriate for adjuvants. One reason why this is really important is that there are so many adjuvants we can use in animal studies to make great vaccines. Most of them are not allowed in humans. And there are actually big differences between the United States and other countries. So the United States basically has like 2 that it lets us use, and mostly it's just aluminum hydroxide. While there are a few others that are approved in other countries, there's one that was like approved in other countries, and it was going to get approved in the US, and everyone was excited because it was going to like really help the US manufacturing vaccine manufacturer industry. And then the year before it was going to get approved in the US, there were side effects in an Italian vaccine that had it. And so then we were all sad because it like went back into trials. And so developing new adjuvants and trying to understand how they work and how we can sort of use them to enhance vaccines without enhancing massive amounts of inflammation at the site is sort of a key thing to think about. You should reread the Tyler paper now. Yeah.
So what's like the threshold for okay side effects and not okay side effects?
So it depends on who you ask. So that is the FDA's decision in the United States or similar groups' decisions in other countries, and they all have their own thresholds.
So which threshold is correct?
That's a great question. There is no, I don't know that there is an answer to, you can answer that what is correct.
Okay, but like in your opinion, what like do you think that the U.S. standard is like too high or too low?
Well, in animals, there's this one called Freund's Complete Adjuvant, CFA. It's the one I always use in problems. I have used that in animals. I have induced anaphylactic shock in those animals because it's such a strong adjuvant. That was not allowed in people. I think that's a good thing. MF9, however, is the one that is sort of they've been trying to approve. I think maybe MF9 might be okay. And I think that there's also this question of what are we trying to prevent against? Do you think that the same adjuvant? Do you think that an adjuvant against sort of a pathogen that's not very virulent, and sort of an Ebola vaccine would be this like would you maybe accept a little bit more risk in your Ebola vaccine? And so there's a lot going on with answering that question. There are two other big ways that we are currently thinking about making vaccines. They are known as recombinant vector-based vaccines or DNA vaccines. In both cases, we know, they rely on the fact that we know of particular antigens that we want the patient to make an immune response against. With a DNA vaccine, we just make a plasmid DNA that expresses the antigen of interest, and we inject it directly into you, and we actually see that protein getting produced, and we see immune responses to it. And so you can imagine that just getting plasmid DNA is real safe. So it's incredibly safe. There are many issues we can talk about with that as well. The other idea is a recombinant vector-based vaccine. And so there we can take, say, the nucleic acid coding for the antigen of interest. So this is Ebola. The antigen of interest is this lollipop-looking one on the outside called GP. And we can actually put that into another microbe that is able to replicate so that that other microbe will now sort of make the one additional antigen. And then we will use this antigen to infect. And so we will then be able to get responses against that antigen. This will be able to replicate and give us all the benefits of replication. It won't need adjuvants, but it will be safe because we use a safe microbe that doesn't cause disease. And so there are lots of ideas about that that had been in trials for a really long time, and there have been some really cool trials for it. And then the 2014 Ebola outbreak happened, and one of these actually got put in in people in the field with very high efficacy, and it has now been fully approved. And so we think that there will be more similar ones approved because sort of the barrier of getting the first one approved has happened, and it's been very safe and very effective. And so that's one place where a lot of people are sort of trying to go right now with design of new vaccine types. There are even situations, a project that I was involved with as a graduate student was that we were taking a harmless microbe and we were actually putting in antigens from HIV, tuberculosis, and malaria. And so in theory, that one infection with that one microbe would act as a vaccine against HIV, malaria, and tuberculosis. And so you can imagine there's some kind of cool ways of dealing with some of these issues. So you guys know that I heart vaccines. I am definitely not getting through all of this stuff, and I'll have to figure out what I'm going to do about that. But the stuff I want to talk about a little bit at the end here is why some people do not get vaccines. And so there are some real legit reasons why sometimes somebody might not get a vaccine. So some people have severe allergies. Some people will have some type of illness or immunosuppression. So there are a bunch of reasons why people won't get vaccines. How many of you, I know the answer to this question, how many of you have had the smallpox vaccine? Zero. Zero of you have had the smallpox vaccine. Unless you have a permanent scar. If you have that scar, and you're like, I might have gotten it, just know that the tuberculosis vaccine, which is given in other countries, gives a similar scar. And so it's most likely the tuberculosis vaccine because they stopped giving the smallpox vaccine in the United States in 1977. And so unless you are a lot older than I thought you were, then you have not, or unless you have experience working with a pox virus in the lab, you have not gotten a smallpox vaccine. I did work with a pox virus in the lab, and so I did have to get one. I always laugh because, in fact, smallpox was declared eradicated 4 days before I was born. Yet somehow I still have had a smallpox vaccine. And so you might think, well, smallpox is really bad. Why don't we get a smallpox vaccine? Why not get it? I apologize about the next slide. So the way that a smallpox vaccine is given is it's given with a bifurcated needle. So it's that little forked needle. They get a droplet of liquid between the needle, and they actually poke around your arm in a little circle. It starts to make a scar, and it's an open wound that you have for about a month. I found this picture online because it's the most realistic to what I feel like mine actually looked like. It looked like I got hit in the arm with a softball for a month. It was very painful, and there were days that I could not get out of bed. It is an infectious virus, and so if you care for it incorrectly, the one that they wrote about that I was terrified of is if you touch it and then touch your eye, you can get vaccinia of the eye. You can also get spread in some cases if you were, say, immunocompromised and didn't know it, or get some type of rash. And so we do think carefully about safety issues and potential vaccine complication issues. And there are legitimate vaccine complications that are seen. You can see some of the complications that are seen after measles vaccine here. 1 in a million people, for example, will get encephalomyelitis after a measles vaccine. However, this is the risk after actually getting the measles, which you can see is much, much higher and much, much riskier, way more possible issues. And so while I am not going to say that vaccines are perfect and that there are no complications, vaccines are incredibly safer than many of the other alternatives.